our professor with biology topic from class 12. Stay tuned and sir, we continue.
Hello students, तो हम लोग वापस आ गए हैं. Today we will study principle of inheritance and variance from class 12. So first let's see what is heredity and what is variation. Heredity means when you get a specific traits from your parents to you, or as in offspring. Offspring means child. And trait, what is trait? Trait is any kind of characteristics. It can be any kind of character, but which is visible or which has some kind of organizational abilities in your body. Okay, those are called as characters or traits. Right. So when a transmission of traits from parents to offspring happens, we call it heredity. It can be through genes on chromosome. We will see that. And then variation. Variation occurs in the individual. Okay. As in case of heredity, you are getting it from your previous generation. But for variation, you are getting it in yourself, as in by various process like genetic recombination, mutation, or in some cases it can be environmental factors as well. So in these parameters like genetic recombination, in genetic recombination your genes are getting recombined, as in various mutation combination is occurring. Through that some kind of changes your genetic code is happening. At the same time, mutation is another spontaneous process we will learn in the further slides. And there are some environmental factors like mutagens and other things. All these can create various genetic changes in yourself. So heredity is from your previous generation. Variation is in the individual itself. We all know Mendel. We have studied it in class 9, 10. Then again, we will study it in this class. Mendel is called father of genetics because he was the first person to realize and then hypothesize the ability of your genes being carried forward to the next generation. By he studied this in plants, specifically guarded pea plants, Pisum sativum. Right, we have already studied this. Why pea plant? I have mentioned it just in case you have some queries. It is very easy to grow. See, most of the time when we are doing science, we choose convenience because you cannot work with something that you don't have availability to, right? So you need something that is easy to grow, clearly differentiable in characteristics. For example, in pea plants, Mendel chose 8 or 9 characteristics like plant height, प्लांट का जो फ्लावर का अरेंजमेंट है वो, फिर he chose the color of flower, color of peas, shape of pea pod, various things. Why? All these characters are very easily distinguishable from the other one. And bisexual flower, as in the same flower you get one female flower, one male flower, not one, multiple, but both the sexes of flower is present. So you can self-pollinate, like this male flower can send its pollen through pollinators into the female flower. So with all these characteristics, you get a very, not very easy, but still much more convenient way of your research. So he chose pea plants due to availability. Mendel's experiment, as we have studied, I will show you again. So he crossed tall plant with dwarf plant. Again, all these characteristics he chose. See, Mendel worked in a older time, so he needed to have some clarification. All these characteristics is just for clarification. Right now, we can choose some very complicated character because we have the ability to perform those experiments. He didn't. So he simply chose tall and dwarf plant. In tall plant, he wrote capital T, small t, both can be tall, and small t for dwarf plant. Let me just explain what TT and TT is. We have seen or studied these these like symbols. What are these? These are genotypes. Whenever we see some kind of characteristics from outside appearance, we call them phenotypes, as in something that is visible to you and to others. But genotype is the code that is inside your cell, in the chromosome, inside the DNA sequence, that is actually ordering you or commenting you to become tall, become short. So in TT or TT or TT, all these are various genotypes for your height. यहाँ पे लिखा नहीं है, but all these are various genotypes for height. Right. Now, let's see this thing. This is a very basic Punnett square I have written for you guys' understanding. I have chose one purebred tall, as in homozygous, and then one short homozygous. Right. Now, all these homozygous plants, TT, capital T, small t, as in pure white, pure tall, and pure short. By this, I have gotten a heterozygous plant. I think you guys already have studied homozygosity, heterozygosity, but let me explain. Homo means same. Hetero means different. Hetero means different, right? Homozygous means when the combination of your genotype is same, as in for one characteristics you have either the same type of genotype, it can be dominant or recessive, whatever it is, but it is same. And in heterozygous you have one dominant, one recessive character. You will definitely show only the dominant character, as we know it is dominant because it is presiding over the recessive character. And when we use this heterozygous plant to self-pollinate itself, we got 1, 2, 3, 4. All 4 of the progeny became all 4 of the progeny. No, there is a little bit mistake. Okay, students, it यहाँ पे थोड़ा changes हुआ है. It will happen like this: capital T, capital T, capital T, small t, capital T, small t, and small t, small t. Right. So we get a equation of 1:2:1. Why 1:2:1? We get one homozygous tall, one homozygous short. All these are 1 and 1. And these two is this and this. These two heterozygous characteristics. We know heterozygosity mein it will become tall, but this recessive gene is hidden inside. So I think you guys understood it. Let's move on. If you have any question, just drop it in the comment box. After teaching you guys, I will see it and I will answer everything. Let's see the aspect of inheritance and variance from Mendel's perspective. What he noted for definition: inheritance is a trait passed from parents to progeny, as I said, and variation is differences in progeny from parents. Means these differences occurred itself in the progeny, right? It occurs itself in the progeny irrespective of your parents' characteristics. Means although you have, not you, for human it is a bit complex, we will see. Maybe a plant has capital T, small t, capital T, small t. Both are tall characteristics, as in traits passed from previous generation to next generation, but I got small t, small t. This is inheritance, but maybe due to some mutation I got this, not because of inheritance characteristics but because of mutation, then it is variation. Okay, we will see further. Causes gene transmission, right? यहाँ पे एक gene है, यहाँ पे एक gene है, we have two different genes and it's getting transmitted in the next generation. It can be by various combination. That is not important which combination. You have to decide which combination based on the question provided to you, but you have to be careful what is going on, because the characteristics your parents have of inheritance, not variation, it will only flow through your next generation, right? So you have to first decide which characteristics you are working on. If you have that, you can easily make a monohybrid or dihybrid cross and you can get the result whatever you need. But always be mindful what you are writing or what the question has for the parental generation. Example यहाँ पे मैंने लिखा है eye color of patterns. Your parent, one of your parent have black eye, one have brown eye, or not brown, one have yellow eye or blue eye. That is, those are recessive characteristics. So maybe or most probably you will get the black eye characteristics from your parent. But height variation in family, it's a polygenic characteristics. Although your main gene that is denoting your height will be important, will be there and will be a determining factor, but there will be multiple various environmental and genetic factor that is coded in your cell, which will make your height a little bit taller or a little bit shorter, whatever. Because we can see कि अगर पेरेंट्स का हाइट एग्जैक्टली है, स्टूडेंट का बच्चे का हाइट उतना नहीं आता, कभी कभी ज्यादा आता है. दिस हैपेंस ड्यू टू वेरिएशंस. Now we will see Mendel's law of dominance. Law of dominance is all characters are controlled by discrete unit called factors or genes, as in TT, TT. Again, these are factors that is determining the trait itself. Small t, capital T. Only these two matters for his height. So these factors will always occur in pair and in a dissimilar pair, as in this pair. In a dissimilar pair, one member, as in capital T is dominant over the recessive, as in this one. Very simple. You can again check the Punnett square. I have written it again. There is a mistake. Let me correct it. This will be capital T, and this will be capital small t, small t. Okay. These are very basic. You have studied it in class 9, 10. Now let's see Mendel's main force law. That dominance, law of dominance, was later Mendel said it definitely. Later scientists have marked it as Mendel law because before Mendel nobody said it. But from Mendel's perspective, he gave only two laws: law of segregation and law of independent assortment. Okay. Let's see law of segregation. What segregation means? Segregation means differentiating. We are separating things. So what Mendel said during gamete formation, we know what gamete is. Gamete is either sperm or ovum that you get from your father and mother. It is always half of your genetic material, right? So if you have, like for example, humans have 46 chromosomes, so we get 23 in the gamete. Now, when you are getting 23 from 46, it will always, each gene will segregate from each other. As in, again, let's go to capital T, small t. I know it's monotonous, but it is the best way to understand. If you have capital T, small t, or capital T, for this height allele, you will always get two different alleles in both the gametes. You will never get 3t and another one is 0, like there is nothing. It can happen, but that is an abnormality. We are talking about normals. See, when you are studying science, you have to always look for the normalcy of the majority things. Whatever exception is there, that is obviously there, but you have to first understand what the baseline characteristics are. Only then you can go understand the exceptions. So what Mendel said, that alleles of each gene will always separate, and each gamete will carry only one allele, as I show you. So these are some important lines. The two alleles in a pair of separate during gametes, they do not blend and are recovered as such in fifth generation. As in, see, यहाँ पे create हुआ, यहाँ से ये लोग अलग हो गए, and then they again they segregate. They mixed up. and law of independent assortment. This is a characteristics from dihybrid course. As in, when you are working with more than one set of alleles, then what Mendel said, they will separate based on their genetic makeup, as I shown earlier in the law of segregation. So they get independently derived or segregated, and this is when happens when one pair of characteristics is independent to other. See, B and A, they separated, B and A, they separated. It didn't happen ki I am getting AA and then I am getting BB. That doesn't happen. Okay. Then it rearranges in the offspring. Obviously, if I cross pollinate between them, you will again get AA, BB, right? And applies on genes on different chromosome. Means you have a chromosome, maybe यहाँ पे characteristics A है, यहाँ पे characteristics B है. So they will separate. Not like this chromosome will get all the A and this chromosome will get all the B. It doesn't happen. Okay. So for Mendel's law and all the process you are gonna perform, you have to remember two things very importantly. One, which characteristics you are working with. जो भी वहां पे जेनेटिक मेकअप दिया हुआ है, इट कैन बी व्हाटएवर स्टूडेंट, डजंट मैटर, बट एग्जाम पे जो दिया हुआ है वो एब्सोल्यूट है, राइट? अगर यहाँ पे आपको देख के लग रहा है दिस डजंट मेक एनी सेंस, इट डोंट हैव, व्हाट यू हैव टू डू इज वर्क विथ दैट one thing. And another thing you have to remember, एक ट्रेट का, सेम ट्रेट का कैरेक्टरिस्टिक्स और एलील ऑलवेज अलग सेपरेट होंगे. वो उसमें आपके लिए मोनो हो, डाई हाइब्रिड हो या फिर ट्राई और टेट्रा हाइब्रिड हो, आपको नहीं आएंगे, फिर भी मैं बोल रहा हूँ ट्रेट्स ऑलवेज सिमिलर ट्रेट का दो डिफरेंट एलील दो अलग जगह जाएगा, अलग जगह ऐसे दो अलग गैमेट पे जाएगा. अगर आपके पास मल्टीपल ट्रेट्स है, आप मल्टीपल ग्रुप ऑफ एलिल्स को एक साथ डिवाइड करेंगे. अगर आपने ये दो बात समझ लिए कि एग्जाम में जो कोड दिया हुआ है वो एब्सोल्यूट है एंड हम हमेशा दोनों सेम ग्रुप के एलिल को अलग अलग गैमेट में भेजेंगे, यू कैन वर्क आउट विथ एनी काइंड ऑफ प्रॉब्लम्स. Now let's see some exception of Mendel laws. Like right now I said that there are exceptions. So let's see these exceptions. Incomplete dominance, right? Right now I show you what dominance is. Now let's see what incomplete dominance. In incomplete dominance, phenotype is a blend of both parental types. Meaning, एक color लेते हैं. Suppose you have one flower with red red, that is the dominant characteristics, and then you have WW. This is a recessive characteristics. Now, if I mix it, it will become RW. And everybody thinks, obviously, if R is there, it will become red. But there is the problem. That is why exception of Mendel laws. In these cases, in these some different situations, this will not become either red or white. Instead, this will become pink, as in intermediate characteristics of both the colors. Got it? So this is what we call incomplete dominance, because dominance is shown but incompletely, because although you are expecting a plant with genetic characteristics of dominant red to get red, but there is also the recessive white, so we are getting a mix of both color as a pink. Got it? Then let me erase these things because I have to write so many things here. And then there is co-dominance. Co-dominance. Co means एक साथ काम करना, लाइक को वर्कर होता है, कोलीग्स होता है, कोहैबिटेशन होता है. तो को मीन्स एक साथ काम करना. जब डोमिनेंट कैरेक्टरिस्टिक्स मैं डी देके लिख रहा हूं एंड द रिसेसिव कैरेक्टरिस्टिक्स विल वर्क साइमल्टेनियस. लेट्स सी हमारा ब्लड ग्रुप का एग्जांपल. आई हैव IA, IB and IO. These are three different genes. Now blood group अगर हम लोग देखें तो we have A blood group, we have B group, we have AB blood group, we have blood. This A and B, these are the dominant characteristics. This O is recessive characteristics. So when we are getting A, obviously we only have two options to get A. One is we have two options to get the genotype A. Sorry, phenotype A. What? One is IA because it is dominant characteristics, or IAIO. Oh, also we write it as IAIO because it is normal. Okay, you can write different things, but don't in the exam. You can remember it, however, but in exam always write it like this, or else they will deduct your number. So we have two options for IAIO and IAIA. All these two will create only A blood group. At the same time, for B, I have IB, IO, and IB, IB, right? And for O is the recessive characteristics, so only one possibility is there. O is the recessive characteristics, only one possibility is there. IO, because it is recessive. But here is the fun. For AB, we see only one possibility, but it is a very important possibility, that is IA, IB. Now let's see. Both are dominant characteristics. According to Mendel, they shouldn't show any characteristics, or either one of them should be the dominant one. But and also from incomplete dominance, we saw the dominant and recessive characteristics get Mendel. So agar IA or IB agar mil jate, so they should have created some weird blood group, but they didn't. What they did, they created AB blood group, as in both the characteristics of A blood group and B blood group is present. Why? Because these two dominant character is working simultaneously, as in they are being expressed simultaneously. In your molecular biology section, you will study one thing called central dogma, where from DNA you get RNA and then you get protein. Always all these traits we are talking about, they are some kind of protein that is being expressed. Now, these IA and IB, these characteristics, they both produce working protein. So we get both the characteristics in our blood. That's why we call it co-dominance, as in co dominant है and दोनों एक साथ dominant है. Then pleiotrope. This is a very simple concept. One gene affects multiple traits. Means अगर आपके पास एक जीन का characteristics है, it can be whatever. Let me write I. This I characteristics is supposed to show, let's say, a protein small I. It will create a protein small I, and this protein will work in maybe scenario A, scenario B, scenario C, affecting these genes in various ways. That is what called as pleiotropy. Again, this is exception of Mendel, because Mendel said one trait is carried out by one gene. Here we can see a single gene is affecting multiple traits. Then polygenic inheritance. Now the opposite of pleiotropy will happen. What happened in pleiotropy? One gene affected multiple traits. Now we will see multiple genes controlling a singular trait. Your skin color, your height, these are all the polygenic characteristics. Why? That's why I said human is a bit complex, because these height, skin color, these are some protein that are determining those factors for you, but those proteins are affected by multiple different characteristics, which you will learn in much higher classes if you continue to study biology. But what you have to remember for your class is multiple genes control one trait, right? Let's move on. Okay, now let's see chromosomal theory of inheritance. We are studying inheritance. We saw the characteristics is being passed from parents to children. But the chromosomal theory of inheritance was proposed by Walter Sutton and Theodore Boveri. What they said? They said chromosomes are the genetic carriers of genetic material. We know, we studied in our lower classes, chromosome is the condensed form of DNA. अगर ये DNA है तो as we know DNA के अंदर हमारे सब जेनेटिक मटेरियल्स मौजूद है. This is the code for you. If you are a software, DNA is the code. Okay. Now, if I take a part of a chromosome which produces a protein, then this part is called as what? Gene. Okay. This is gene, a functional part of your DNA that is creating a protein. There are many non-functional part of DNAs. Okay, those don't produce protein or functional protein, so they are not called as gene. Gene should always produce some kind of functional units. So chromosome obviously a carrier of DNA. As I said, chromosome is made up of condensed DNA. DNA is the carrier of gene. See, you have to connect those things and then study. Just रट्टा मत मारा करो, तो फिर भूल जाओगे एग्जाम तक पहुंचते पहुंचते. They occur in pair. Obviously, we studied chromosome occur in pair. Not this, that one. These are two different homologous chromosomes which went through replication to create pairs. Right? These pairs are called as sister chromatids. Segregation of chromosome occurs during meiosis anaphase 1. When we study cell cycle, we will see details में meiosis, mitosis क्या है, उसका 4 भाग है, हाँ, prophase, metaphase, anaphase, telophase. Now you have to remember for inheritance is ये जो homologous chromosome है sister chromatid, let me erase it. ये जो chromosome है, ये homologous chromosome का sister chromatid, they will separate, okay? They will separate into singular strand like this during anaphase, and they will move to form two different gametes. Meiosis is the process of creating gametes. We will study in cell biology. Stay connected, okay? And this theory linked Mendel abstract factor to physical structures seen in cell. What is that? Why abstract and why factors? Let's see, okay? Oops. मेंडल ने जो फैक्टर्स के बारे में बोला था और ट्रेट्स, he said this happens because I can see the phenotype, and he was right. But as scientists, we shouldn't always accept some people's word for it, even though they have logical hypotheses. So as further science improved, we tried to see what these factors actually are. As in, something is making you tall, something is making you short, something is making you dark colored skin or white skin, eye color, every characteristics. But if you can see the characteristics physically, that means there are some kind of physical aspect to the factors as well. That is what Sutton and Theodore Boveri did. They gave this abstract concept of factor a physical structure, that is your chromosome. Then let's see linkage and recombination. Linkage क्या होता है? Tendency of genes present close together in the same chromosome to be inherited together. Meaning, let me draw a chromosome again. See, this is a chromosome, and as I said, अगर दो sister chromatid है आपके पास, it will separate during meiosis anaphase 1. Okay. Now, if this has like here, यहाँ पे अगर दो आपके characteristics है, यहाँ पे और दो characteristics है. Suppose let's name them A, B, C, D. Now this A, B, and C, D, they will try to move together always. In case of cell division, meiosis, anaphase 1, all these concepts are based on cell division, anaphase, and mostly meiosis. Okay. In some cases, you will see later these are not getting inherited together, but that is not a characteristics of cell cycle. Meiosis, those are happening because of again some exceptional situation, some disease or those kind of situation. In normal cases, in the majority of cases that we study as a normal baseline hypothesis, they will always separate together. Okay, they will always separate together. So that is what our scientist T.H. Morgan named as linkage, as in these genes are linked together. Okay, you are with me? they are linked together. At the same time, recombination, generation of non-parental gene combination due to crossing over during meiosis. See, I said difference happens. For example, let me erase that. यहाँ पे आपका एक क्रोमोसोम है, यहाँ पे आपका एक दूसरा क्रोमोसोम. Now, what I said, they are supposed to separate. Now, what happens if they cross-link with each other? So, what will happen is कि, let me draw a red color here. और यहाँ पे एक और रेड कलर दे देता है. Sure. And ये पोच देते हैं. Let make it a bit simple for you guys. तो हमारे यहाँ पे है, this is our recombination is happening. So अगर recombination हुआ ये A1 जीन के अंदर, क्रोमोसोम के अंदर, तो क्या होगा? It will work like that is what happens is कि see यहाँ से जो रिकॉम्बिनेशन हुआ उसके वजह से ये पोर्शन कट के यहाँ आ गया एंड ये पोर्शन यहाँ चला गया. सो नाउ chromosome A has both the red mark part and chromosome A1 has neither red mark part. These are some, what should I say, these are some process by which generation of non-parental genes, as in the genes that you didn't get from your parents or inherit from your parents, they cross over and then they create some different combinations. All these combination creation, and previously we studied the mixing of various gametes, all these for the need of variation, because when we will study evolution we will see the concept that variation give rise to evolution, as in if you are not different from all the member of your species you will not get naturally selected. We will see those things in further classes, so stay tuned. Okay, रहना है साथ में. हाँ बच्चे, so recombinant frequency is used to make genetic maps. What is that? Again, for example, you have various linked genes and recombination. You can calculate their distance. मतलब जीन नंबर 1 से 2 से 3 से 4 5 6 कितना दूरी में है. Okay, that you can calculate. You don't have the mathematics yet, you just have to know the process. So I'm telling you, math we will study in few further classes. So another interesting thing about linkage is we can calculate linkage. Linkage as in linkage is inversely proportionate to distance between genes. Why? ये दोनों जीन 1 and 2 जितने पास होंगे, it will be that much harder to separate them. For example, your best friend that you sit with in your class, as much as you make noises and your teacher want to separate you, you guys always find an excuse to sit together the next day. That is what happens to the genes also. They are that much closely situated that without any external forces in your body, they will not independently separate in ever. So that is why it is inversely proportionate to distance between genes. If the genes are like that, there are many chances of centromere breakdown and recombination occurring for making 1 and 2 separate. Okay, and the distance of linkage, ये जो है recombination frequency का distance, this is written as unit cm. Okay, cm means centi-Morgan. Morgan है, this is the scientist who first discovered linkage, तो उसके सम्मान में हमने नाम रख दिया centi-Morgan. It is not centimeter. Okay, be careful. The c is small in the as in lowercase word and m is uppercase. I have seen your textbooks, this is mentioned. So if they were asked what is the unit for the linkage or unit for the distance between two genes, write down either centimorgan, or if you write cm, make sure to write small c, capital M. Don't make these silly mistakes. Now let's see some different type of inheritance. Till now we have studied inheritance from a autosomal perspective. Autosomal perspective. Autosomal means the characteristics or the chromosomes and genes that all are like denoting your physical characteristics. Now let's see some sexual characteristics. Sex linked inheritance. We know in humans, we will study only human genetics, sex linked inheritance. We have XX for females and we have XY for males. Oh, also these symbols, memorize them, okay? Because for exam you may see these as the question में सीधा सीधा ये symbol दिया हुआ है and they didn't mention male female. So make sure you are aware of these symbols. They are denoting, this is denoting female, let me write, and this is denoting male. Okay, we can remember it as in plus because मम्मियाँ हमें गोद में लेती है. This is why it is a plus sign, and ये आपका मेमोराइज करने का ट्रिक्स है, बस कुछ भी तरीके से बस याद रखिए. So let's get into the topic. Sex linked inheritance में क्या होता है? Sex linked inheritance में you get some X linked traits, means these characters or these traits are connected to the X chromosome itself. Okay, they are connected to the X chromosome. For example, hemophilia हुआ, color blindness हुआ. These are characteristics that is linked with the X gene, means Y में इसका कोई प्रेजेंस ही नहीं है. They are not, sorry, minus होगा. They are not present in the Y chromosome at all. Okay, they are not present in Y chromosome. X में इनका एक presence है. At the same time, some Y-linked characteristics are there. These are called as Hollandric characteristics because of the scientists who invented it. So Y is a Y gene के अंदर क्या होता है? Male में होता है बस Y gene, we know. So these characteristics are, these Y-linked traits are male specific. Okay. Male specific, as in these characteristics will never occur in the female group. Never. Because females do not have any Y. There is not a resemblance of Y. Why we will learn. So X-linked characteristics can be present in both male and female. But there is another fun part. For female, these X-linked characteristics is a double genetic characteristics है. Okay. क्यों? क्योंकि यहाँ पे दो एक्स क्रोमोसोम प्रेजेंट होते हैं फीमेल में. सो दे values for every X-linked characteristics, but for male they only have one X chromosome and one Y. Y का जो पार्ट है उसमें तो कोई X का कैरेक्टरिस्टिक्स है नहीं, तो डाटा बची एक. तो ये जो X लिंक है फीमेल, मेल मैं लिख दे रहा हूं, यू गाइस नोट इट डाउन. मेल इज मोनोगैमस for X-linked traits. Okay, male are homogamous for monogamous for X-linked traits, meaning they only have one gamete or one chromosome, whatever you call it, one gene, one chromosome hypothesis. So it can be one gene, one chromosome, whatever you call it, but they only have one copy of it. These are the things that they will ask you in for exam, because they don't need to know how much you have rectified your topics. They need to know how much you have understood. Even this is very important for NEET also, that X-linked traits are monogamous for male and bigamous for or digamous for female. Y-linked characteristics only occurs in male. Now let's see sex dependent or influence trait. What is sex dependent or sex influence trait? Meaning this is a characteristics that is autosomal. but it is influenced by X or Y, as in a characteristics, a gene trait that is present in your autosome is influenced by your sexual characteristics. For example, one thing we have seen many times, that is male pattern baldness. You can see many males have this part of their hair receding and the hairline getting back and back as they age. Why does it happen in mostly in males? See, for that we have to know the male pattern baldness का characteristics. मेल पैटर्न बॉल्डनेस का डोमिनेंट जीन है कैपिटल बी एंड रिसेसिव ना सॉरी I forgot the spelling of recessive. Recessive characteristics is small b. Now, if we make a Punnett square with it, capital B into small b, we get capital B small b. Now we got building that as P1, right? If it is P1, let me erase some part of it so that I can write again. Okay. Now let's see for this B capital B small b as in the heterozygous character, इसको अगर हमने पेरेंटल जीन पेरेंटल जनरेशन 1 पकड़ लिया तो क्या होगा? सेल्फ क्रॉस होगा. यहाँ से we get only 4 combinations, that is capital B capital B, capital B small b, capital B small b and small b small b. Okay, now let's see. यहाँ पे मैं एक चीज लिख रहा हूं, ओके, फिर आपको समझ में आएगा. इफ मेल एंड इफ फीमेल, ओके, नाउ लेट्स सी व्हाट हैपेंस इफ मेल एंड इफ फीमेल. कैरेक्टरिस्टिक तो सेम ही रहेंगे. व्हाट हैपेंस? ओके, लेट्स सी. इन बोथ केसेस दे विल हैव हेयर बिकॉज ये जो बी कैरेक्टरिस्टिक्स है, दिस इज डोमिनेंट. लेट मी राइट बी dominant and small b recessive. Okay, now if male and if female both में अगर ये हमारा जेनेटिकल कैरेक्टरिस्टिक्स एज इन जीनोटाइप कैपिटल बी है, तो दे बोथ विल हैव sufficient amount of hair, as in they will not have male pattern baldness. But अगर यहाँ पे क्या small b हुए, then both will be bald, whether it is male or female, both will be bald, right? Because this is a recessive characteristics. Now the fun part occurs here. If it is a heterozygous characteristic where one dominant and one recessive character is there, male will be bald, but the female will have hair. Why? The presence of Y chromosome determines whether you have testosterone or not. Testosterone अगर आपके पास है कि नहीं, this is determined by Y का presence, right? Now, if Y is present, as in testosterone is present, it will influence these heterozygous characteristics, and for male, this capital B or the dominant character will be suppressed, and you will get bald males. But if there is no testosterone, then it will if there is no testosterone, it will be a female. Definitely female individuals do not have testosterone. Then this suppression of small capital B will not occur, and the dominant characteristics will show and you will get here. sex dependent influence trait, as in the character is definitely autosomal, but it is getting influenced by your sexual characteristics, as in presence and absence of Y. Okay, let's see the other part. Now, sex limited trait. Okay, sex limited trait means it is limited to one sex, मेल फीमेल, इनमें से किसी एक के पास ही होगा. फॉर एग्जाम्पल, द एबिलिटी टू प्रोड्यूस मिल्क थ्रू मेमरी ग्लैंड, that is only possible for female. It is not like there are some autosomal factor, there are some genetic factor, nothing. Female embryo से programming ऐसे होता है, only they will have working mammary gland and they will create milk. So these are sex limited characteristics. Okay, so we got three kind of sex inheritance. One is sex linked inheritance, means either X chromosome or Y chromosome में characteristics होगा and we will work with that. Then there is sex dependent traits. These are autosomal traits. Let me write क्योंकि आप लोग तो भूल जाएंगे. This is the characteristics is autosomal, but it is influenced by your sex chromosomes. The presence and absence of X and Y वो character को either suppress or uplift करेगा. Then there is sex limited traits. These are only specific for either male or female. You cannot see these characters simultaneously. Got it? Let's move on. Sex determination. We will study a few different type of sex determination to understand the differences between the process of sex determination. See, sex determination in general, most of the animal in the world ecosystem or world biome is sexually dimorph. Sexually dimorph means one individual will become male and another individual will become female. But this dimorphism, how is it created? For humans, we have studied multiple times. XX are for females and XY for males. How? We will see later. इसको थोड़ा अभी के लिए रख देते हैं. Now let's see insects. For insects, we see a XO and XX characteristics. Mean male are heterogametic, like human also. In human, we have XY for male, right? In plant, in insects, we have male के लिए हमारे पास है XO and female is XX. Simple, not much is there. Now for birds, bird में males are homogametic. Okay, males are homogametic, ZZ, and ZW. The heterogamous situation is for females. These are the differences you have to remember for the exam. They will ask ZW system में which individual, which sex has the homogamous situation, which sex has the heterogamous situation. Remember them, learn them, answer them. All these characteristics like XO, ZW system, XO system और हमारा वाला XXXY system, all these are determined by your permutation combination. If you have W, you will become female. If you have Y, you will become male. If you have O, you will become male. But there are some environmental factors sometimes. In reptiles, mostly crocodiles, then turtles, these reptiles live in a amphibian nature. Okay, they have the access to both water and ground. So their sex determination evolved with time into a much more environmental based development rather than genetic based development. Okay, in crocodile, sex is determined by environmental temperature. अगर 30 डिग्री सेंटीग्रेड 30 डिग्री सेंटीग्रेड से, अगर 30 डिग्री सेंटीग्रेड से इक्वल या नीचे गया टेम्परेचर, टेम्परेचर एज इन एनवायरमेंटल टेम्परेचर, यू विल गेट फीमेल एग, माने एम्ब्रियो, एग का अंदर जो एम्ब्रियो है इट विल डेवलप इनटू फीमेल. एंड इफ इट द टेम्परेचर इज मोर देन इक्वल टू 34 डिग्री सेंटीग्रेड, मतलब अगर एनवायरमेंटल टेम्परेचर 34 डिग्री सेंटीग्रेड या उससे ऊपर गया, देन यू विल गेट मेल्स. Remember these very nicely. If you need, you take screenshot. For turtles also, take screenshot of this thing because you need this. They will ask some kind of question involving this, and don't mess it up. So these two are environmental. Environmental factor determines your sex development. Now there is a very fun thing called genic balance theory, meaning obviously there will be some balance between genes. In Drosophila we can see genic balance theory. What happens is you have autosome and you have X chromosome, right? Now autosome by X chromosome, इसका जो ratio है, that will determine whether you get male or female. If it is greater than equal to 1, you will always get female. If it is less than equal to 1, you will get male. It is as simple as that, but not simple, because neither autosome nor X chromosome is determining the sex. This is a ratio, means in some cases if the A chromosome ka number is aneuploid or polyploid, as in number got increased or decreased, the ratio of sex changes in the population itself. Now let's see sex determination in human. Okay, for human let me write something. That is, in human men, most important thing you have to note is यहाँ इससे लिखते हैं sex determination in human is by default female. Okay, by default, FAULT, default female, means if there is no X chromosome, it will always become female. Every embryo that has been created throughout the human history was supposed to be female. Only and only if Y chromosome is present, then they will become male. Else, every chromosome, every zygote, every individual is programmed to become a female individual. If there is a different code that is provided by Y, then will they become male. This is a very important concept. Okay, remember it properly. By default, human are programmed to become female. If you have Y chromosome present in your genetic system, then you will become male. How? Let's see that. In Y chromosome, there is one thing called SRY. What is SRY? SRY is called as, यहाँ पे लिख देते हैं, sex determining region of Y. Okay, SRY, sex determining region of Y. This is a part of your Y chromosome. In Y chromosome, we have two different shape of arms. Okay, those are very less minute important topic. You can go through your textbook. But this SRY part is present in your Y chromosome. This SRY part is a gene. Okay, remember the difference between gene and protein. A gene is written as capital letters. If I write SRY, then it is the SRY protein that is formed from SRY gene. These are very minute nuances, but these are the things that most students make mistakes of. This capital SRY, as in the SRY gene, has a codes multiple protein, but one of them is TDF, testicular determining factor. Testicular determining factor is the factor that will determine your testes. So if there is no TDF present, like it is written here, absence of TDF leads to default female reproductive tract development. Meaning, if there is TDF, only then male reproductive tract will be developed. Take a screenshot of it, do whatever you want, but remember it. That female is the by default characteristics of human embryo. Everything will become female, as in human. Every human will become female unless there is TDF present. Got it? Let's move on. Mutation. Let's study mutation. Is the source of variation. What is source of variation? Source of variation means you are an individual A. I am individual B. We got these characteristics of A and B from our parental generation, P1, say P1. Through our life, by living and getting exposed to various environmental and genetic factors, we became A.1 or B.1. That means not major things change, but minute changes happen throughout our life, in our body, in our gene itself. If that happens, that is what causes variation. Okay. Now let's see mutation as a source of variation. What is the definition of mutation? Remember some part of it properly, very proper. Mutation is a sudden, spontaneous, and heritable change in your DNA. Meaning, it will occur very randomly and spontaneously. You cannot, or nothing actually, no human factor, no anthropogenic factor can determine or predict or influence mutation. Mutation occurs suddenly, randomly, spontaneously, and it is a heritable change, meaning as mutation occurred, your DNA may, it will put some information in your DNA that will be permanent, and this permanency will be heritable, as in it will be heritated into your F1, then further down the line. Okay. So mutation is a sudden, spontaneous, and heritable change in the DNA sequence. If you remember this much, it should be like, it should be written as it is in your exam if definition of mutation comes and it is the right one. Mutation can be of two major types: gene mutation, chromosome mutation. Mean, as I said, chromosome is itself gene because chromosome contains DNA, DNA contains gene. But when we are saying gene mutation, meaning a specific जीन के अंदर कुछ चेंजेस आया है. A gene, it was supposed to be AATTCG, AATTCG, AATTCG. There were some changes made that made it CCTAGC. So that is gene mutation in a specific gene. Chromosomal mutation occurs in a broader aspect, as in it affects the whole chromosome as it is. मतलब अगर 23 पेयर जो है उसमें से 22 प्लस 1 ए बन गया, मतलब किसी एक क्रोमोसोम में चेंजेस आया. ओके, so then it will be called as chromosomal mutation. Mutagens are physical or chemical substance that causes genetic mutation. Okay. You can write it down, take a screenshot of it, whatever you want to do. Well, let's move on to the mutation part. It is very interesting. First, let's see chromosomal mutation. क्रोमोसोमल म्यूटेशन के अंदर दो तरह का चेंजेस हो सकता है. स्ट्रक्चरल चेंज, एज इन द स्ट्रक्चर ऑफ द क्रोमोसोम एज इट इज चेंज्ड, और इट कैन बी नंबर चेंज, मतलब नंबर ऑफ क्रोमोसोम 1 था it multiplies, or neither of them is present. It can be anything. So that is why we call it polyploidy or aneuploidy. Poly as a word means multiple, meaning the number of chromosome that is supposed to be 46, it became 47, 48, 49. Okay, then it is polyploidy. Aneuploidy में क्या हुआ? Again 46 will change into 45, 44, like that. मतलब the number of chromosome you are supposed to have decreased or increased. Now let's see the structural changes. Okay, this is very intricate, so follow me as I say things. First we see deletion. Again we have a chromosomal structure. Let's paint this part as gene A. Okay, from deletion we get something like this. The whole part of the gene A became silent, or it was deleted from the gene set, from the chromosomal segment. So the chromosome became A negative for gene A, means the gene A that was present in the chromosome previously, it is not there anymore. The gene, a part of the structure of the chromosome itself got deleted. Okay, डुप्लीकेशन में क्या होता है? You have a gene with you have a chromosome with gene A duplicated, meaning it became both are A. Okay, it's not like it duplicated and changed its characteristics. It is same as it is the gene, just it duplicated. It can also duplicate here, not necessarily only have to be here. It can duplicate in the different arm of the chromosome. Okay, so this characteristic causes your gene to multiply in a certain number, so we call it duplication. Then comes inversion. Okay, see it nicely. I am marking a bigger part for you to understand. Okay, it is like that. Through inversion, what will happen is see the structure of the chromosome removed itself and then rotated in on its axis, creating the upside down structure, and then it attached itself into the chromosome itself. Okay, so what happened is, if I write it like that, you will understand nicely. Maybe A, B, C, D. Okay, and when inversion occurred, it became A, D, C, B. So this portion turned over in on itself. Okay, so that's why it is inversion. Inversion meaning यहाँ ऐसे पेन था, हमने घुमा दिया. This is inversion, a rotation of 180 degree. Okay, then comes translocation. Translocation में क्या होता है? Translocate होता है, as we can see. There are two chromosomes. Okay, one has this gene part. What occurs is, in case of translocation, ये chromosome A है, ये chromosome B है. Okay, the red part from chromosome A detached itself from the chromosome and got attached into a separate chromosome, or this is one type of translocation called as Robertsonian translocation, where, remember it, okay, properly understand it, when a part of a chromosome remove itself and attaches to another chromosome, it is called as Robertsonian translocation. And the other type of translocation is, again, there is a part of DNA which is red, chromosome which is red. Translocation will occur and the chromosome का जो ये part है, it attach itself in the same chromosome but on the different arm. P, Q. It was P and Q. See, it was in the P section, it came to the Q section. This is as called as translocation. Okay. So these are all the structural and number of changes in the chromosome which will cause your chromosome to mutate. Deletion means negative हो गया, minus हो गया, कोई एक part of DNA is removed, part of chromosome is removed. Duplication, part of chromosome is duplicated and then added in on itself. Inversion 180 degree का shift होता है. Okay, gene was like this, it became like this. Translocation, either you will remove yourself and connect to your same chromosome on a different arm, or you will connect yourself in a different chromosome altogether. Polyploidy means increase, polyploidy is increase, aneuploidy is decrease. Now let's study gene mutation. हमने अभी तक पढ़ा chromosomal mutation. Chromosomal mutation में chromosome as it is getting changed. Now we will move ourselves to the genetic sequence. For example, A, T, G, C, T, A, C, G. This is a section of your DNA that I picked. Maybe it contains some genetic information. It is random. Okay, do not say कि sir Pratyush sir told there is a gene like that. It is random. But let's say this is a genetic sequence. There can be two types of mutation: substitution, frame shift. Substitution means something is substituted by other, and frame shift means this is a frame, I moved the frame like this. This frame move हो रहा है. This is what is called as frame shift. Let's see. Let's first see substitution mutation. Okay, transition, transversion. What happens is कि जे substitution mutation में only single nitrogenous base pair का changes होता है. Okay, single one only one nitrogenous base pair will change. Then we will call it substitution mutation. यहाँ पे हुआ suppose. Okay, so now there can be two type of substitution. One is A, T, T, C, T, A, C, G, and another can be A, T, C, G, T, A, C, G. What happens is यहाँ पे G got replaced by T and C. If it is replaced by T, as in the same group of purine, अगर प्यूरिन के साथ सब्स्टिट्यूट हुआ या फिर अगर पिरिमिडिन के साथ सब्स्टिट्यूट हुआ, then we will call it transition. As in, I am transitioning from one purine to another purine, or one pyrimidine to another pyrimidine. Okay, we are changing the group of the nitrogenous base stays same, only the code changes. प्यूरिन के साथ सब्स्टिट्यूट हो गया, पाइरिमिडिन के साथ सब्स्टिट्यूट हो गया. तो वी विल से सब्स्टिट्यूशन इज ट्रांजिशन हुआ सब्स्टिट्यूशन. ऑन द अदर हैंड देयर इज ट्रांसवर्शन में क्या होता है? ट्रांसवर्शन में प्यूरिन इज रिप्लेस्ड बाय पाइरिमिडिन और पाइरिमिडिन इज रिप्लेस्ड बाय प्यूरिन. लेट मी राइट, बस आप लोग तो भूल जाओगे. प्यूरिन is substituted by a pyrimidine or pyrimidine is substituted by a purine, then we will call it transversion, as in a transverse property nitrogenous is found. There ट्रांजिशन हुआ नाइट्रोजनस बेस का प्यूरिन से दूसरे प्यूरिन में ट्रांजिशन हुआ, यहाँ पे एक प्यूरिन से दूसरे pyrimidine में ट्रांसवर्शन हुआ. ओके, नाउ लेट्स सी फ्रेमशिप म्यूटेशन. फ्रेमशिप म्यूटेशन के लिए अगेन वी नीड अ सीक्वेंस. फॉर एग्जांपल, also what is frame shift? For to know that we need to know what frame is. We know a codon is written like this: AUG, GUC, ATC, with 3 different nitrogenous sequence. This is called as reading frame, meaning one codon, another codon, another codon. We study codon with a three different nitrogenous base. We study it as one codon. So when frame sheet occurs, this codon का जो स्टडिंग फ्रेम है या फिर रीडिंग फ्रेम है वो चेंज होता है. कैसे मैं दिखाता हूं. AUG, ATC, GUC, these are 3 different codon situated at a DNA sequence. ओके, 5 प्राइम टू 3 प्राइम में ये कोडोन्स है. अभी अगर मैं यहाँ पे मैं एक नाइट्रोजनस बेस ऐड कर दिया तो क्या हुआ, ऐसे हुआ. ए मैंने सपोज यहाँ पे एक टी ऐड कर दिया, फिर उसके बाद रहा बाकी सब कुछ सेम रहा. यू जी ए टी सी जी यू सी 3 प्राइम 5 प्राइम. नाउ इफ वी वांट टू स्टडी द जेनेटिक कोडिंग, पहले हम पढ़ रहे थे AUG, नाउ वी आर स्टडिंग ATU, GAT, CUG, CGU एंड हमारा वो बन गया इससे 3 पेयर. तो हमारा जो कोडोन का रीडिंग फ्रेम है, हम जो कोडोन 3 के सेट में पढ़ रहे हैं, दैट रीडिंग फ्रेम इटसेल्फ चेंज्ड. So the codon sequence changed. So the protein sequence changed. That is why we call it frame shift. The reading frame of the codon itself is changing. Not only the codon के अंदर एक substitution हो गया, नहीं, the codon frame, codon का reading frame itself is changing. Okay, it can be insertion or deletion. Meaning ये जो मैंने दिखाया, this is insertion. Okay, क्योंकि एक T हमने add कर दिया. Or the other thing I can do is A G A T C G U G. यहाँ पे भी अगर हम लोग पढ़ने चाहे तो AGA TCG UG. मैंने यहाँ से जो ये U रिमूव कर दिया, उसके वजह से U का जो डिलीशन हुआ उसके वजह से भी रीडिंग फ्रेम चेंज हो गया. दिस इज वे ऑब्वियस. लाइक हमने नॉर्मली अल्फाबेट लिखा है ABCDE, आप लोग पढ़ रहे हैं ABCDE, सर ने लिखा है ABCDE. बीच में मैंने C हटा ही दिया, बीच में से मैंने C हटा दिया. अभी पढ़ो, अभी आप पढ़ोगे तो ABDE ऑब्वियसली. तो आपका जो रीडिंग फ्रेम है वो इटसेल्फ चेंज हो गया. या फिर मैंने, आप फिर से पढ़ रहे थे, सर ने लिखा है ABCDE, वेरी गुड, सर ने बीच में से घुसा दिया, सर ने बीच में से एक T घुसा दिया, तो क्या हुआ, तो भी सेम चीजें हुआ. आप पढ़ रहे थे ABCDE, आपको अभी पढ़ना पड़ा ABCTDE. तो आपका रीडिंग फ्रेम में आइदर डिलीशन हो रहा है या फिर इंसर्शन हो रहा है, आइदर और. आपने जो आपका जो स्टडी का सीक्वेंस है, आपका जो कोडोन का रीड का सीक्वेंस है, वो खुद चेंज हो गया. दिस इज व्हाई वी कॉल इट फ्रेम शिफ्ट म्यूटेशन. योर रीडिंग फ्रेम इटसेल्फ इज चेंजिंग. नाउ लेट्स कम टू द जेनेटिक डिसऑर्डर्स. ये अभी तक हमने पढ़ा कि मेंडल ने इतने सारे थ्योरीज दिए जिनका म्यूटेशन होता है. हमने पढ़ा कि कैसे वेरिएशंस आते हैं. व्हाई ऑल दीज थिंग्स टू नो हाउ आवर जीन फंक्शंस, and when we know how our gene functions, then we can see the disorder that we are getting from those genes. कि एक जीन का काम करने का तरीका होना चाहिए था ऐसे, उसका काम करने का तरीका हो गया ऐसे ऐसे, राइट? सो दिस इज व्हाई स्टडीइंग नॉर्मैलिटी इज इंपॉर्टेंट। पहले भी आपको जो बता रहा था, एक्सेप्शंस वी कैन ओनली लर्न व्हेन वी ऑलरेडी नो द नॉर्मैलिटी। अगर नॉर्मैलिटी में चेंजेस नहीं आए तो वी कैन नॉट कॉल इट एक्सेप्शंस। तो मेंडेलियन डिसऑर्डर है, क्रोमोसोमल डिसऑर्डर है। मेंडेलियन डिसऑर्डर मतलब है, हमारा मेंडेल का क्या मतलब मोरल ऑफ द स्टोरी मेंडेल का क्या था कि यू विल इनहेरिट कैरेक्टर independently from your parent to yourself, and those characters can be formed through various permutation combination which is independent with each other. This was mostly what Mendel tried to say. If I have to say it in one sentence, it is you will get characteristics from your parents, and those characteristics can make any type of permutation combination independent. Mendelian disorder matlab disorder that are inheritable. Okay, characters those are inheritable. मतलब ये कैरेक्टरिस्टिक्स, ये डिसऑर्डर्स जितने सारे हैं, यू कैन इनहेरिट फ्रॉम योर पेरेंट्स. क्रोमोसोमल डिसऑर्डर मीन्स there is a disorder changed in your DNA, sorry, in your chromosome itself. मतलब आपका जीन का कोई चेंजेस नहीं हुआ. आपको अगर आपको सपोज क्रोमोसोम नंबर 20 उसके अंदर जैसे जीन्स रहने थे वैसे ही है, उसके अंदर जो सीक्वेंस रहना था वैसे ही है, शायद ये क्रोमोसोम खुद ही है नहीं, या फिर 20 का 3 कॉपी आ गया आपके बॉडी में. These are chromosomal changes. Obviously, these are also inherited. But when we talk about inheritance, we mostly means that it is inherited by genetics. As in, as you reproduce, your gamete itself carries the information through its gene. Okay. Now let's see. Hemophilia is an X-linked. Hemophilia is X-linked recessive, affects blood clot factor. This character is linked in X chromosome itself. I was telling you guys about sex-linked traits, X-linked traits. This is a X-linked trait, hemophilia. The characteristics of hemophilia, as in the genes, is itself present in X chromosome. Okay. So अगर X क्रोमोसोम में डोमिनेंट है तो एवरीथिंग इज फाइन. अगर रिसेसिव है तो वो हिमोफिलिया का इंफॉर्मेशन कैरी कर रहा है. अगर दो रिसेसिव बन गया या फिर एक रिसेसिव और एक वाई जीन बन गया तो हिमोफिलिया आ गया. व्हाट हैपेंस इन हिमोफिलिया इज, आपने ब्लड क्लॉटिंग पढ़े होंगे 11 में, आपका जो फिजियोलॉजी टॉपिक था उसमें ब्लड क्लॉटिंग था. तो वो जो टॉपिक है उसमें आप देखेंगे दो फैक्टर है, फैक्टर 8, फैक्टर 9. दीज आर वेरी प्राइमरी एंड इंपॉर्टेंट फैक्टर्स दैट कॉजेज योर ब्लड टू क्लॉट. ये दो फैक्टर में गड़बड़ी आ जाती है, ये दो फैक्टर का जो जेनेटिक सीक्वेंस है वो घट जाता है, तो ये दो प्रोटीन प्रॉपर्ली प्रोड्यूस ही नहीं हो पाता. तो क्या होता है, ब्लड क्लॉटिंग ही नहीं होता. सिकल सेल एनीमिया इज ऑटोसोमल रिसेसिव डिसऑर्डर. और इसमें क्या होता है, इसमें एक कोडन है GUG ओ GAG में चेंज हो जाता है. तो हमने जैसे पढ़ा पहले ही, सी इन्वर्शन ट्रांसलोकेशन, नो सॉरी, सी ट्रांजिशन ट्रांसवर्शन, दैट इज व्हाट हैपेंस, राइट. पॉइंट म्यूटेशन होता है, एक सिंगल पॉइंट में, एक न्यूक्लियोटाइड के एक पॉइंट में चेंज आ गया. तो ये कोडन कोड करता है वैलिन को, ये कोडन कोड करता है ग्लाइसिन को. आपका बनने का बात था एमिनो एसिड, वैलिन बन गया ग्लाइसिन, तो काम कुछ हुआ नहीं. Color blindness is also an X-linked recessive characteristics. Phenylketonuria is recessive autosomal disease. All these disease you memorize go through multiple times for your exam. This will come as MCQ objective type question in your board, and this is also important for NEET because these are physiological questions, so you need to remember these questions for NEET also. Chromosomal disorder, Down syndrome, Klinefelter syndrome, Turner syndrome, all this chromosomal factor is due to this number changes. Okay, क्या होता है ट्राइसोमी ऑफ 21, मतलब 21 जो क्रोमोसोम है आपका आपका जो 21 क्रोमोसोम है उसका कॉपी रहना चाहिए था 2 कॉपी, लेकिन आपका बना 3। तो अगर 3 कॉपी बन गया तो क्या हुआ, यू हैव 1 एक्स्ट्रा सेट ऑफ क्रोमोसोम, तो आपका गड़बड़ी हुआ, 46 रहना था 47 हो गया। यहाँ पे क्लाइनफेल्टर सिंड्रोम में you have XX and then 1 Y, it will become male. As I said, if there is Y present in any scenario, there can be 100 X's before Y, but if 1 Y is present, it will definitely become male. तो 47 क्रोमोसोम हो गया मेल में, रहना था 46। या फिर टर्नर सिंड्रोम, उसमें एक एक्स क्रोमोसोम है ही नहीं, राइट, इट वाज सपोज टू बी एक्स एक्स, बट एक क्रोमोसोम है ही नहीं, तो हो गया एक्स ओ, रहना था 46 क्रोमोसोम, रह गया 45। एक्सेप्ट दैट देयर इज आल्सो एडवर्ड सिंड्रोम, व्हिच कंटेन्स 13 क्रोमोसोम का ट्राइसोमी होता है। ओके, सॉरी 18 क्रोमोसोम का ट्राइसोमी होता है, क्रोमोसोम नंबर 18 में ट्राइसोमी आ गया, क्रोमोसोम नंबर 18 में ट्राइसोमी आ गया। ऑन द अदर हैंड देयर इज पटाउ सिंड्रोम, जिसमें क्रोमोसोम नंबर 13 का ट्राइसोमी आ गया। ट्राइसोमी मींस 3 सोम एज इन 3 क्रोमोसोम, मोनोसोमी मींस 1 क्रोमोसोम। We always are supposed to have 2 chromosomes, but if you have tri, tetra या फिर mono, that means number of chromosome you are supposed to have, you don't have. This is pedigree chart. Take a screenshot of it. Remember whatever is written here. These are the code which is agreed upon by biologists everywhere, biologists throughout the world. Whenever one geneticist writes this, we all know that he means a normal male. Normal means, यहाँ पे normal मतलब नॉर्मल से की बात नहीं हो रहा है, यहाँ पे नॉर्मल मतलब हो रहा है कि इट इज अ नॉन डिजीज्ड इंडिविजुअल। ओके, female, unspecified sex, all these are for non-diseased individuals. These are affected individuals or diseased. Okay, then they are deceased. Disease और deceased में मिलाना मत, हाँ, डिसीज मतलब मर गया, हो गया, इसका बात खत्म। डिजीज मतलब ही इज सिक। दिस इज मेटिंग मतलब मैरिज हुआ, देन दे प्रोड्यूस न्यू ऑफस्प्रिंग। दिस इज कॉन्सैंगुनियस मैरिज, दिस मीन्स मैरिज बिटवीन रिलेटेड इंडिविजुअल्स। रिलेटेड इंडिविजुअल्स मतलब फर्स्ट कजिन, second cousin, in fact it can be brother or sister as well. This causes genetic depression. मीनिंग जेनेटिक डिप्रेशन का मतलब क्या है, मतलब आप जितना इनब्रीडिंग करोगे, आप जितना रिलेटिव और मतलब रिलेटेड इंडिविजुअल्स के साथ ऑफस्प्रिंग प्रोड्यूस करोगे, योर जेनेटिक इंफॉर्मेशन विल गेट फनेल्ड इन एंड इट विल क्रिएट इनब्रीडिंग डिप्रेशन. सो दैट इज नॉट अ आइडियल सिनेरियो फॉर एनी ऑफ अस. You can see some specific example of consanguineous marriage in इंग्लैंड का जो रॉयल ब्लड फैमिली है, दैट दोस मैथ्स वी विल डू, दोस पेडिग्री मैथ्स, दे आर ओनली यू कैन सी consanguineous marriage in a context that you will understand. This is heterozygous condition. Obviously we have 2 chromosome, तो अगर एक डोमिनेंट हुआ एक रिसेसिव हुआ तो आपके पास हेटेरोजाइगस आ गया, दोनों नहीं हुआ तो होमोजाइगस है, आपको पता है. दिस इज हेटेरोजाइगस बट सेक्स लिंक्ड. this is heterozygous but autosomal trait, this is sex trait. These symbols you have to remember. अभी आप अभी रिमेम्बर कर लोगे अच्छा है, वरना आपको एग्जाम के बाद घर आके देखना है क्यों गलती हुआ है, लेकिन ये आपको रिमेम्बर करना है. ये है स्टिल बर्थ मीन द इंडिविजुअल डाइड एट बर्थ. ओके, दिस इज फेटर्नल ट्विन एंड दिस इज मेटर्नल ट्विन. ओ ये है ना किसी तरह से गलती हो गया, लेट मी राइट. ये है ट्विन का सिंबल. इसके एंड में क्या है वो मैटर करता है. अगर दोनों फीमेल है तो फिर दोनों फीमेल है, दोनों मेल है तो फिर दोनों मेल है, या फिर दिस ऑल्सो पॉसिबल. दिस इज फेटर्नल ट्विन मीनिंग दो डिफरेंट गैमेट से दो डिफरेंट इंडिविजुअल पैदा हुआ है, इसीलिए इनका सेक्स अलग है. यहाँ पे इट कैन बी दो डिफरेंट इंडिविजुअल दो डिफरेंट गैमेट से पैदा हुआ है, लेकिन बोथ का अंदर सेम गैमेट था इसीलिए सेम सेक्स हुआ है, या फिर एक सिंगल गैमेट बाद में दो डिफरेंट इंडिविजुअल में डिवाइड हुआ है. ओके, आइडेंटिकल ट्विन्स कैन नॉट एवर बी फेटर्नल, एज इन इनके बीच में कभी भी दो डिफरेंट जेंडर नहीं आएगा सेम ट्विन में. आइडेंटिकल ट्विन का आइडेंटिटी का सेम है बिकॉज दे आर मेड फ्रॉम द सेम जाइगोट. द जाइगोट throughout the process of development they differentiate and creates two different zygote. They contain the same information. Fraternal twin can be in both, in any combination, male male, male female, female male. The identical twin always will be the same gender. मैं फिर से एक बार रख रहा हूं, आप लोग देख लो, स्क्रीनशॉट ले लो, कुछ भी कर लो. Now these questions, okay, we will come back after a 5 to 10 minute break and see these questions, and we will solve these questions with our other biology educators for you guys. Okay, be right back in a few seconds after taking a little bit of break, and let me drink some water. तो हम लोग फिर से आ गए आपके साथ कुछ क्वेश्चंस डिस्कस करने। हम मेरे साथ पल्लव सैलरी है, we have introduced him like yesterday also. No. Okay. This is so I'll add another indicator. So I'm Pallav. So let's see the question. Yeah. Certainly. Actually, we are having some technical issues with our other mic, so we are gonna share this mic today.
So yeah, the question is, what will never be father's blood group if the mother has blood group B and child has blood group O? So let's see the answer.
So first we have to see something. If the child has blood group O, O is recessive character, that means it will be IO, definitely, right? Now, if the child has IO means it got one IO from his mother and one IO from his father, right? We are clear up to that? Now, if we see this and mother has B blood group, that means mother definitely will be IO, right? Now, if the mother has IO and it get one IO from mother, then another IO came from father. So the possibility of father will be IO will be definitely there, except that it can be IA, obviously, it can be IB also, or it can be IO also, because this can be variable, because this is what we definitely get into the child from father. So if he has one IO and another this or this or this as a 2.1, 2.2 or 2.3, then the impossible phenotype of father's blood group will be IA, IB. Why? Because there is no IO, so father can never be AB. He can be A, B or O, as we saw, IO, IA for A, IO, IB for B, and IO for O, but can never be AB. Let's move on to the next question. Oh, sir want to say something?
क्योंकि ये वाला बहुत इंटरेस्टिंग एक क्वेश्चन है और बहुत बार आया है एग्जाम में, तो उन लोगों को थोड़ा और एग्जांपल ये ब्लड ग्रुप रिलेटेड मैथ्स में करवाने से थोड़ा सा ठीक रहेगा.
We'll see. And also for this type of repetitive question regarding one specific topic, you definitely have to join our mentorship, okay? Because there we will not only discuss some model or some pre-IQs, we ourselves will make some repetitive questions from the same group with different type of topics, so that you can see the differentiation of the same topic in these questions. मतलब एक ही क्वेश्चन के टाइप टॉपिक में कितने सारे डिफरेंट टाइप ऑफ क्वेश्चंस आ सकता है. ओके, जी सर ने बहुत इंपॉर्टेंट बात बोला है आप लोगों के लिए, क्योंकि यू हैव टू ट्राई आउट द differences, only then you will understand this properly. So follow us and join our mentorship program for this kind of differentiation of the same topic. Okay sir, today ऐसे भी हम लोगों ने बहुत वक्त ले लिया, थोड़ा और दो चार क्वेश्चन देख लेते हैं. In sickle cell anemia, glutamic acid is replaced by valine. Which codes for valine? I said you GUG. Okay, यहाँ पे अगर A रहता तो it will code for glutamic acid. Then phenotypic ratio of dihybrid cross in F2 generation, it will always be 9:3:3:1. We have done dihybrid cross, we know what happens. It will be always the phenotypic ratio. Genotypic ratio will be 1:2:2:, 1:2:2:, 1:2:2:, 1:2:2:, 1:4:1:, 1:1.
So question can come.
These you cannot remember, okay? I know this by memory because I have done that many dihybrid cross throughout my study career. So you have to, only way to know and learn Mendelism is
Actually, this mentorship program we have designed in a such a way that every student gets 100% attention from us. Okay, that's why we are doing 1:5 mentorship. That is one mentor against 5 students
per batch.
per batch. Yes.
There will
Eh,
be many batches. So please,
Exactly. We have lots of mentors. Need not to worry. There will be rush. So please, please, please hurry up. Enroll right now. And we will give you premium videos for this. Those will be recorded one for your chapter revision. Actually, what you are doing now in home, you are practicing now. You are doing revisions. So that is done by us. वो रिवीजन हम लोग कराएंगे आप लोगों को 1:5 के बैच में हम लोग आप लोगों को पूरा चैप्टर का रिवीजन कराएंगे थ्रू प्रीमियम वीडियो रिकॉर्डेड लाइव सेशन में हम आप लोगों का डाउट क्लियर करेंगे हम लोगों का आप लोगों का असेसमेंट करवाएंगे आप लोगों का PYQ पैटर्न क्वेश्चन डिस्कशन होगा फॉर अपकमिंग बोर्ड्स सो प्लीज डोंट बी लेट हरी अप
there will be mock test also.
एग्जैक्टली sorry
It's
I forgot
important.
the series of mock tests will also be there
So it's important for practicing as much as possible.
exactly
Please join us.
please. Thank you for this session. But stay tuned. There is our next session for Maths Class 12. Hello students, BookmarksEducare is offering you a free webinar on 25th and 26th October for upcoming board exams.
Boost your preparation in physics, chemistry, maths and biology with experts guidance.
For class 10 students, the topics covered will be chemical bonding, circulation, volume and surface area, electricity and magnetism.
And for class 12 students, the topics covered will be principles of inheritance, solutions, continuity, differentiability and higher order derivatives, and modern physics.
Time is ticking. Enroll right now. Surprises are waiting for you. Hello students, BookmarksEducare is offering you a free webinar on 25th and 26th October.
For 10 students, the topics covered will be chemical bonding, circulation, volume and surface area, electricity and magnetism.
And for class 12 students, the topics covered will be principles of inheritance, solutions, continuity, differentiability and higher order derivatives, and modern physics.
Time is ticking. Enroll right now. Surprises are waiting for you. Hello students, BookmarksEducare is offering you a free webinar on 25th and 26th October for upcoming board exams.
Boost your preparation in physics, chemistry, maths and biology with experts guidance.
For class 10 students, the topics covered will be chemical bonding,